Today's lesson is going to focus on meiosis and really why sex and sexual reproduction are so important to the continuation of life and genetic recombination. So one of the first important things to know is that sexual reproduction is what produces genetic variation. It's when you have members of the same species being a little different, and it's what allows us to keep up with changing environments. Remember, asexual reproduction gives you organisms that are exactly the same, and for environments that don't change very often, that isn't a big deal. But for environments like where we live and where you see these fish would live, it's a big deal to be just a little bit different because it allows for a little bit of variation that allows some organisms to live longer, reproduce more, and get more of their genes into the next generation. With every generation, meiosis and some meiotic recombination of the genetic makeup gives us a new combination of genes for that species. So what is meiosis? It is a reduction division process that occurs only in the gamete-producing cells. Remember that gametes are things like egg and sperm. So let's talk about a little bit of vocabulary here for just a second. So meiosis, remember, is a reduction division process, and what it produces are haploid gametes. Haploid basically means half. So what it's going to do is take an organism and it's going to reduce the number of chromosomes it has in half so that when these two things come back together, their offspring, or in this case the zygote, is back to the normal diploid number of chromosomes. For example, you have 46 chromosomes in each of your cell. That is your diploid number. However, the egg and sperm that were put together to create you are a haploid number of chromosomes. Therefore, there are only 23 in each one. So what we're going to talk about today a little bit is how these cells are actually formed. Meiosis begins very similar to mitosis. In this case, we're going to actually have S phase where we double the number of chromosomes. So remember, I said that you have 46. During S of interphase, that number is going to double to 92. And those chromosomes are going to be held together in the center with their centromere just like they were during mitosis. The next phase is prophase 1. So something that will happen here is called synapsis. Synapsis is when homologous chromosomes, so here's another big term that's important. Homologous means same. So this is the same chromosome and their sister chromatids line exactly up gene by gene, forming what's called a tetrad. So basically, you have your chromosome, its sister chromatid, and your second chromosome and its sister chromatid. Remember that your chromosomes appear in pairs because you received one from your mother and one from your father for each and every set of your chromosomes. So if this is chromosome number 1 and after S it duplicates, these two are what will line up right here to form a tetrad. Something that can occur during prophase 1 that is definitely worth talking about is called crossing over. This is when DNA is exchanged between homologous chromosomes. So as the homologous chromosomes will intertwine, a portion of one can actually break off and exchange with a portion of the other. So what this creates is more variation in traits. So if you were to inherit this chromosome, you're not just getting the genetic information from that original chromosome from your parent, you're actually getting a little bit of one chromosome and a lot of another, which gives you even more variation. So the next phase is metaphase 1. At this point, we take those tetrads that were created during prophase 1. Remember when the homologous chromosomes paired up and lined up, and we actually begin to line them up at the metaphase plate. So this is very similar to mitosis. Remember, meta means middle, so that's where these guys are going to be lining up. And the way that these assort themselves, so the way that they line up is very much left to chance. And so this is a really big deal. So this is part of the reason why we never really know which chromosomes are going to go into the next generation. It all depends upon the way they line up on this metaphase plate before they get pulled to the other side during anaphase. Which leads us right into anaphase 1. This is where the homologous chromosomes, or those tetrads that were created, are simply pulled apart to opposite sides of the cell. Now we still have 92 chromosomes. Nothing was separated completely. We still have those copies, and they're all still in one cell. During telophase 1, we're actually going to get 2 cells formed. Now, we still consider this to be haploid even though there's 46 because we haven't finished with meiosis. So we have 2 new cells during telophase 1, but we are not done. We now have meiosis 2 to go on to. Now, this is going to go through the same steps, but in this case, instead of being prophase 1 and metaphase 1, we're going to have prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, this is going to take place in both cells simultaneously. What you'll notice is that there is no interphase, really, between meiosis 1 and meiosis 2. So DNA is not replicated. So we're going to have 46 chromosomes in each set of these. And prophase 2, we're going to get our spindle forming, and our sister chromatids are paired up the way that you see them here and the way that you see them here. During metaphase 2, those sister chromatids are going to line right up along that metaphase plate. So in the middle of the cell, they're going to get attached to from the spindle. During anaphase 2, we're going to get those sister chromatids. Remember, they were in their X shapes. We're going to rip them apart to opposite sides of the cell. And telophase 2 is going to come in and start to separate those 2 different cells completely apart. So now, because both cells were being separated, we have 4 haploid, remember haploid means half, cells. And in the case of humans, you would have 23 chromosomes in each of those cells. And all 4 cells, and this is big, have different genetic information. Each of these cells now has the potential to go on and either fertilize or be fertilized, creating new life. So what is the difference? We have 2 different kinds of gametes that can be created. We have sperm that can be created through a process called spermatogenesis. Genesis means to create. Or we could have eggs that are produced during a process called oogenesis. Now, during spermatogenesis, we get an equal division of the cytoplasm. So let's look here. We have our first meiosis occurring. Down here, we're getting our second set, or meiosis 2, to occur, and you get an equal division here during telophase 2, and so all 4 of those cells can become sperm cells. In oogenesis, it's a little different. We have unequal cytoplasm division. So during this very first, so meiosis 1, where you end up with the 2 cells, one of them is much bigger and gets much more cytoplasm. During the second division, that first one still maintains that large amount of cytoplasm, and there ends up being these 3 very small cells called polar bodies and 1 large egg. During spermatogenesis, the whole process will begin at puberty and continues throughout the life of the male. You're going to get several 100 million sperm being produced every single day. Because remember, every time that cell goes through meiosis 1 and then meiosis 2, all 4 of those cells can become functional sperm. So you get way more of those than you do eggs. Now, once the sperm are formed, they move into the epididymis, where they actually mature a little bit and they're stored. The process is a little different, remember, with oogenesis. So you get that first division into the unequal division of cytoplasm, and then each of those divides again. So only one of them really becomes functional. The ovary contains many follicles composed of developing egg surrounded by an outer layer of the follicle cells. Each egg begins oogenesis as what's called a primary oocyte. At birth, a female will carry with her a lifetime supply of these developing oocytes, each of which is just hanging out in prophase 1. A developing egg is released each month, beginning at puberty until menopause. Basically, menopause occurs when you run out of eggs, and it's a total of about 400 to 500 eggs. So there are some actual sizes here of a few eggs just to kind of show you in comparison. So this is a chicken egg, this is a frog egg, and up here is a human egg. What occurs next is the magic of life. It's fertilization. Out of those hundreds of millions of sperm each day, one of them might possibly fertilize an egg. And that's what's happening right here in this picture. They're all attempting to get in and fertilize that egg. Those 23 chromosomes in the sperm from spermatogenesis, which all occurred through meiosis, and the 23 chromosomes in the egg, which happened in oogenesis, which is the process of meiosis again, come together to form a 46 chromosome zygote, and life continues. Here's just a chart with a really good comparison of mitosis and meiosis. Remember that mitosis is going to happen in body cells. So mitosis happens in your toe, whereas meiosis happens only in the egg and sperm producing cells. So the first cell may look very similar. You have those duplicated chromosomes, but during mitosis and during meiosis 1, we really start to see a big difference. At the very beginning, crossing over can occur during prophase 1. That does not happen during mitosis. We also see those homologous pairs line up at the equator. And one of the biggest differences we see is 2 sets of divisions during meiosis as opposed to mitosis. And at the end of mitosis, both of these cells are exactly the same. Whereas at the end of meiosis, they have half the number of chromosomes and they are completely different from one another. Here's another comparison chart, but here we have the differences in words between mitosis and meiosis. Take some time to read over these and maybe even write them down.